Greetings, general science students. Here we are. We're going to finish up Module 16 today. Okay, very good. Go ahead and open your lab book to pages 77, and you'll go through 79. And hopefully I'll have all those blanks that are in there in the right places where you can fill them in. Okay, so we're finishing up the nervous system, and today we're going to be looking at the peripheral nervous system, and especially the sensory division of the peripheral nervous system. So we'll be looking at our senses today. There are 5 senses. They are vision, sight, hearing, taste, touch, and smell. And then there's a 6th that we've added, and you may not think of it as a sense. We've added it because it is registered in a sensory organ. And the sensory organ which it is registered in is the ear. Any guesses on what that sense is? Oh wait, let's go through taste, smell, sight, sorry, I didn't forget about this, touch, hearing, and here it comes, balance. All right, turns out we balance through the work of what's happening inside of our ears. When we get to that, I'll explain that to you. It's pretty fascinating. It seems like there's actually more devoted to balance than to hearing in there. How do we taste food? All right, so the brain, here's our first blank to fill in. Our brain receives messages from the taste buds. Now, the taste buds are on our tongue, and we have 5 taste sensations, and here they are: sweet, salty, sour, bitter, and they just added this one, savory, also known as umami. The best way to describe that one would be Chinese food, something like that, Worcestershire sauce. Now, this picture over here on the right, it says a map of the taste receptors. So it turns out that we have taste buds all over our tongue. And in the old days, they used to think that we mostly tasted bitter back in the back and sour on the sides and salt and sweet. We don't believe this is true anymore. So this has been pretty much debunked. We can taste everything all over, but it is true that along the edges and tip of our tongue, we do have more taste buds, and therefore we can taste it better there. So here we've done away with this. We've decided it is not true, but it is still kind of fun to taste things on different parts of your tongue to see if what you think about that. Okay, so our taste buds. The taste bud is like this, all right, and it looks a little bit like the bud of a flower. It has a little hole. So this is your tongue right here, this part right here. It has a little hole in there, and it's called the taste pore. That's the hole, right? And then the taste bud is down here. And at the top there are these little gustatory hairs. Yes, there are hairs on your tongue. And those receive chemicals that can dissolve in your saliva, and when that happens, we can take that and send that message. So it's called a chemoreceptor. It takes that chemical down to the nerve right here, and the nerve goes to your brain, and it tells you a couple things. One, it tells you which one of those flavors it ends up in, one of those 5 flavors right there. Furthermore, your brain will also have a memory. So for instance, if you're tasting salty, buttery popcorn, you'll know, oh, that's that salty flavor there. And I remember it. It's the taste of popcorn, you see. So your brain's pretty cool that it can remember those tastes as well. If we look at it a little closer, this is kind of gross. Here's the pores, very, very up close. There's that little gustatory hair, right? Those hairs right in there that are receiving those messages, okay, and sending those to our brain. This is also a close-up view because it turns out our taste buds are a little different. We have our filiform taste buds that look like this one, and our fungiform taste buds that look like this one. So many of the filiform don't really have any role for tasting except that they hold stuff down in there, right? So there's place for liquids to sit. And the fungiform are really responsible for for having all those chemoreceptors. Furthermore, there are people out there, you might know some, maybe they don't know, this is the deal, that cannot really take really strong flavors. They just don't like really strong flavors, bold flavors. They like things kind of bland. They might be what's known as a super taster. And a super taster, you might think, awesome, that'd be so great to be a super taster. Oh, not so much. Yeah, because therefore when you try new things that are really flavorful, you may not like them. So I think I have one of my sons is one of these because he just won't even, try some stuff because he just does not like the taste. Those people have more of the fungiform taste buds, and so they're super tasters. I'm not a super taster, and I'm happy because that means I like a wide range of different flavors of food. All right, moving on to the sense of smell. It turns out that the sense of smell is tied pretty closely to the sense of taste. They kind of work together. All right, so it helps us taste our food. We call this the olfactory sense. All right, so olfactory sensory cells are inside of our nose. These cells signal the brain, and the brain determines the smell, and I'll show you how that happens. Sometimes those trigger memories, like, I know what that smell is. So in other words, your brain remembers the smell, but then there'll be other memories, like you might smell something and say, oh, that reminds me of that summer when we were at the beach, or something like that. So it not only has memory as I know what that is, but it might evoke, emotional memories for you as well. All right, so inside your nasal cavity, you can see, so you take a breath in through your nose here, and at the very top of your nasal cavity right there are these specialized cells. So you see the bone, right here is the bone of your skull, and we have these nerve endings actually coming down through the cribriform plate right there and into these cells right here. Okay, so we have these cells are they are releasing some mucus. So you see here the mucus layer. So these are specialized cells, epithelium, to release that mucus on there. So the mucus is covering this part right there, and then these olfactory hairs, once again the hairs, are coming down there. And when a chemical comes in and dissolves, I think I might have a video on this, so we'll watch that. It dissolves in this, and it takes that message back to our brain. Let's see if I do. Yeah, let's watch this video. There's one little part in here that talks about smelling urine. I'm sorry. Yeah, sorry about that.
It's the first sense you use when you're born. 1 out of every 50 of your genes is dedicated to it. It must be important, right? Okay, take a deep breath through your nose. It's your sense of smell, and it's breathtakingly powerful. As an adult, you can distinguish about 10,000 different smells. Here's how your nose does it. Smell starts when you sniff molecules from the air into your nostrils. 95% of your nasal cavity is used just to filter that air before it hits your lungs. But at the very back of your nose is a region called the olfactory epithelium, a little patch of skin that's key to everything you smell. The olfactory epithelium has a layer of olfactory receptor cells, special neurons that sense smells, like the taste buds of your nose. When odor molecules hit the back of your nose, they get stuck in a layer of mucus covering the olfactory epithelium. As they dissolve, they bind to the olfactory receptor cells, which fire and send signals through the olfactory tract up to your brain. As a side note, you can tell a lot about how good an animal's sense of smell is by the size of its olfactory epithelium. A dog's olfactory epithelium is 20 times bigger than your puny human one. But there's still a lot we don't know about this little patch of cells, too. For example, our olfactory epithelium is pigmented, and scientists don't really know why. But how do you actually tell the difference between smells? It turns out that your brain has 40 million different olfactory receptor neurons. So odor A might trigger neurons 3, 427, and 988, and odor B might trigger neurons 8, 76, and 2,496,678. All of these different combinations let you detect a staggeringly broad array of smells. Plus, your olfactory neurons are always fresh and ready for action. They're the only neuron in the body that gets replaced regularly, every 4 to 8 weeks. Once those neurons are triggered, the signal travels through a bundle called the olfactory tract to destinations all over your brain, making stops in the amygdala, the thalamus, and the neocortex. This is different from how sight and sound are processed. Each of those signals goes first to a relay center in the middle of the cerebral hemisphere, and then out to other regions of the brain. But smell, because it evolved before most of your other senses, takes a direct route to these different regions of the brain, where it can trigger your fight or flight response, help you recall memories, or make your mouth water. But even though we've all got the same physiological setup, 2 nostrils and millions of olfactory neurons, not everybody smells the same things. One of the most famous examples of this is the ability to smell so-called asparagus pea. For about a quarter of the population, urinating after eating asparagus means smelling a distinct odor. The other 75% of us don't notice. And this isn't the only case of smells differing from nose to nose. For some people, the chemical androstenone smells like vanilla. To others, it smells like sweaty urine. Which is unfortunate, because androstenone is commonly found in tasty things like pork. So with the sweaty urine smellers in mind, pork producers will castrate male pigs to stop them from making androstenone. The inability to smell a scent is called anosmia, and there are about 100 known examples. People with allicin anosmia can't smell garlic. Those with eugenol anosmia can't smell cloves. And some people can't smell anything at all. This kind of full anosmia can have several causes. Some people are born without a sense of smell. Others lose it after an accident or during an illness. If the olfactory epithelium gets swollen or infected, it can hamper your sense of smell, something you might have experienced when you were sick. And not being able to smell anything can mess with your other senses too. Many people who can't smell at all also can't really taste the same way the rest of us do. It turns out that how something tastes is closely related to how it smells. As you chew your food, air is pushed up your nasal passage, carrying with it the smell of your food. Those scents hit your olfactory epithelium and tell your brain a lot about what you're eating. Without the ability to smell, you lose the ability to taste anything more complicated than the 5 tastes your taste buds can detect: sweet, salty, bitter, sour, and savory. So the next time you smell exhaust fumes, salty sea air, or roast chicken, you'll know exactly how you've done it, and perhaps be a little more thankful that you can.
Okay, yeah, so the sense of smell is the good one. It does help with our sense of taste. So remember, we only have the 5 tastes, but 10,000 things we can smell. So they absolutely help one another. And like it said in the video, if you've ever been sick and you're all stopped up, you can notice how things just don't taste as good. Yeah, so that's pretty cool stuff. All right, now we get to the main act here. We're looking at vision. We will talk about hearing and ears and stuff too. But I love the eyeball. We're going to dissect our eyeball here in just a little bit. Okay, so we have 2 eyes. That seems kind of obvious. But it's very nice for us that we have 2 eyes, because what it helps us do, it gives us a certain amount of depth perception, and also it will accommodate for what we'll look at in a minute, something called your blind spot. Since you have 2 eyes, we can overcome the blind spot. Okay, so we can see here the parts of it right here. The sclera is this white part, also called the bulbar conjunctiva. We have the iris right here, and then the pupil. So this iris is actually a muscle, and it opens and closes our pupil. Okay. We also have tear ducts right here that keep the eye moist at all times. What do you think the eyelashes are for? And furthermore, even the eyebrows. Do you know what that's for? It's all about keeping stuff out of your eyes. You've all had something in your eye before, and it hurts, right? And it's like you can't do anything until you get that taken care of. And so the idea is that our lashes and our brows will help to keep things away from the inside of our eyes. Okay, so let's go over to this side right now. Here's that sclera. That's the white of your eye. I want you to see how that sclera extends all the way down right here. And we're going to see in our dissection here that, interestingly, this part of the sclera is clear. It's in the same material, however. Very, very cool. All right, so you see kind of a bulge right there. In fact, if you close your eye and put your finger over your eyeball, you can kind of feel that little bit of a bulge right there coming out there, if it doesn't hurt too badly. All right, so then we get in here, and the eyeball itself is filled with fluid. It's called aqueous humor, vitreous humor that's in there. All right, so here the clear cornea, light will come through. The light is going to come through the hole, which is our pupil, and it's going to be focused by this item right here. This is the lens, okay? The iris is the muscle right here that's going to open and close depending on how much light we want to come in through the lens right here. All right, so this part here, the iris is continuous here, right? So the ciliary, oops, I want to go back. The ciliary muscle right here, including the iris. Okay, so light will come through, and remember, this is filled with fluid, and it will cause there to be, the image to be focused. Now we can go on to this one. All right, so in the back of our eyeball, there are specialized cells in an organ called the retina, okay? So in the retina, which basically is pasted on the back of the inside of each eyeball, it has cells in it like this, okay? So we have cones that look like this green one here, and rods. Now there's a lot more rods than cones, and that has to do with because the rods are for just taking in any kind of light. Okay, so the rods will detect light and versus dark, basically shades of gray, if you will, and the cones will register color. In order to see color, we have to have lots of light. You may have noticed in the evening how things just sort of aren't as colorful, right? The sun's starting to set, and things just sort of lose their color. That's because they don't have any as much light shining on them. The more light that's shining on something, the more colorful it will appear, because it's all about reflection of light. And it's those cones that pick that up, and the rods will pick up the shades in there. All right, so as a light stimulus reaches the retina, these cells, which are called photoreceptors, are able to interpret that light. All right, I believe this is the only place where we have these photoreceptors. So this is the inside of the eyeball, so the retina right here. And you can see it's going through all these pathways to get to those specialized cells right there. So there's a cascade of biochemical events. And when I say that, you should say, oh, no, it's really difficult. It is very difficult to understand because without each step along the way, vision will not take place. There have been scientists out there who have studied this biochemical pathway of vision and have become believers in it. Because after looking through this, there's no way they figure that this could have happened accidentally. Even if it happened over millions of years of evolution, think about it. If each one of those steps in this biochemical pathway is going to take 1,000,000 years to get there, that means I have several million years of an organism who has no vision. All right, so that vision is going to be one of our chief abilities for survival, isn't it? Think about an animal that's getting away from a predator. Seeing them will be one of their chief ways of avoiding them, right? And so therefore, like I said, these scientists, after studying this, figured this had to be created ready to go. It had to all be there. All those steps had to be present. Otherwise, survival would have been difficult, if not impossible. All right, we're back to the eye here. Good. I'm glad we have this. If we were together, this would be up on the board, right? And I would take a marker and I would draw through here. So light travels in a straight line. So I would have light coming in through the pupil, and the lens will now bend it a little bit, okay? And then it will focus it on the movie screen back here, which is your retina. Okay, so it's this movie screen type situation. There's one little hole in it right there where the optic nerve comes in. Okay, it's called the optic disc. And that little spot right there in this eye, you have no vision right there. Okay, we call that the blind spot, which we'll get to soon enough. All right, so notice all the blood vessels right there. The blood vessels are feeding all those cells that are in the retina, all those rods and cones and those neurons and things that are in there. So when we are looking at something, light is coming through our eye. And let's say that we have something over here. I wish I had a picture over here to show you. I would draw like a Christmas tree, and I would show you that the top of the Christmas tree will come a straight line like this, and the bottom of the Christmas tree straight line like this, and then the straight lines everywhere else. But anyway, the image would appear upside down on our retina. That's just a rule of optics, okay? And that is passing through the lens like that. The light actually will cause the image to turn upside down. You can do this if you hold out a magnifying glass and hold it and look at somebody, they're upside down, right? So same thing happens. So the question is, how come we don't see everything upside down? Well, our brain just flips it for us, just flips it so we don't go crazy thinking everything's upside down, right? Okay, so then depending on the amount of light that comes in, we will, so for instance, if it's a very, very bright day and you're out in the sunshine, then your pupil actually gets very, very small because it doesn't need to be very big to allow the maximum amount of light in there. But yet if you're in a very dim room, let's say you go to the movies, right? So you're sitting at a movie theater. I believe the movie theaters have opened back up, if I heard the governor right yesterday. So you can now go to a movie theater, and it's dark in there, but let's say it's the middle of the day. You finish the movie, you walk out the door, and bright sunshine. What do your eyes do? I mean, you just go, oh, it almost hurts. It probably does hurt. And what's happening is that ciliary muscle that has your iris in it, that is going to squeeze down very quickly. So it's very big. So your pupils open wide when you're in a dark room. And then when the light comes in it quickly, whoop, that closes and causes that little bit of pain while it's closing up right there. Now you can do this if we were together. What I would have had you do was to take a mirror and shine a flashlight into your eye, and you could see your pupil just close like that. Okay, that is a part of your nervous system that is doing that. It's recognizing the amount of light that's coming in. It's kind of fun to watch. I'd let you do it to me too if you want to shine it in my eye. Furthermore, another thing that's interesting, if I shine a light in my left eye, my right eye will also constrict. Okay, so that's a neurological test. Some people can tell if there's been brain damage to somebody if they shine a light in their left eye and their right eye doesn't constrict down and get smaller the same way, then they might have a brain injury or something. So that's interesting to know. All right, so in a few minutes I'm going to show you our dissection of this. Most of you have your eyeball, and so you'll be doing your own dissection of this right here. But you see there's all this like physics that go into the focusing of the image right here, because the light comes through here and it focuses over here at the fovea centralis, sort of right in front of it, almost like it's going to shine that image onto that screen. Okay, very cool. Now this is something that we would be doing if we were together. I don't know if it's going to work right now, okay? This is locating your blind spot in one of your eyes. So what you're going to do for this, you're going to look at the screen. I don't know if you have your phone or your computer or whatever, but if you cover your right eye with your right hand, okay, and you look at the screen, with just your left eye, you can see the plus mark and you can see the dot, right? Now I want you to just look at the plus mark. Can you still see the dot even though you're looking at the plus mark? Yeah, right? You can see it over there with your peripheral vision. Okay, so keep just looking at the plus mark and bring your face closer to the image. And there's going to come a point, if it works right, where you won't be able to see the dot anymore. Okay, so like I said, I'm not sure if this is going to work on the screen like this. Maybe it's too big. I thought it worked for me yesterday. It doesn't seem to be working today. But go ahead if you want to try to pause the presentation right here and try it a couple times. You could take like a 3x5 index card and put a dot on one end and a cross on the other end and try it. So remember, you're going to cover one eye and you're going to look just at the little plus mark, and as you move it in, that dot will disappear. I mean, if you look over at it, the dot will still be there. You understand what I'm saying? You're only looking at the plus mark. Does that make sense? Okay. Then you could try the other eye, and see if it works. Okay, so give it a go and see if that works. This is an optical illusion. Okay, so stare at this for a minute. And for me, it looks like it's moving. Does it look like it's moving to you? Okay, so I will tell you it's not moving. All right, it's just the way the lines are drawn. Our eyes are trying to take all of that in. It actually kind of tires your eyes out and gives you a little bit of eye strain if you look at it too long. So I won't leave it up there too long. It might be interesting if you pause it at this point to see if it looked different when you pause the deal. I don't know. See if it still looks like it's moving. Here's another one. This looks like if you kind of move your eyes around to the different circles in there, it kind of looks like they're rotating. Okay, so move your eyes around a little bit. Like look at the bottom row and move around, and it sort of looks like they're moving. They're not moving, but some people look at it and say, I don't see the movement at all. Okay, I see it. It looks like they're rotating to me. All right, this one also rotating slowly. To me, it looks like it's rotating slowly to the right. Okay, and it's just a way of tricking our eyes by loading a whole lot of information onto our retina at once. And like I said, it makes your retina tired. This is just a trick of the eyes. So does that have, how many prongs are there? 2 or 3? Follow that around and see, and it's nonsensical, right? It doesn't make sense. Oh, yeah. I have another one of those here. So this is much more obvious or easy to trick you, I guess you could say, when it's up on the big screen right there, because I can ask you, which is the green circle larger or the purple circle larger? And you might guess the purple circle, but I would put a piece of paper up to it. You can do that and just show they're exactly the same size. The green circle and the purple circle are the same size. They're influenced by what's around them. Those big black circles around the green circle make it appear smaller versus the tiny circles around the purple make it look larger. These are optical illusions. Here, this one kind of just made me laugh because are we looking at his profile? Or are we looking at him staring straight at us? Yikes. If you want to stop these at all, you certainly can. Okay, moving on to the sense of touch. Sense of touch allows us to sense changes in our environment, to respond to those changes. If we touch something hot, we move away from it, right? We can detect slight change, wind, temperature, things like that. And this is due to receptors and nerves that work together in the skin. Okay, some skin cells have more receptors than others. For instance, our hands are very sensitive to the sense of touch, obviously, because we're going to be using them a lot for touching things. So in your book, there's an experiment in there, I guess on page 415, which we could have done. Sometimes we don't have enough time, and I skip this one anyway. But it will be where you're basically taking it's what's called 2-point discrimination, two toothpicks, and seeing if you can feel both of them or just one of them. So when they're so close together, sometimes they're a part of your skin. You actually can only feel one poke. Oftentimes, though, we have you do this with a partner so you're not watching it happen, because sometimes when you see it, it makes you think you're feeling it too. Does that make sense? Yeah. All right, so what kind of sensory organs do we have in our skin? So we know we have our dermis and epidermis. We know that, right? This is our integumentary system. And so we have some free nerve endings. Where are my free nerve endings? I thought this was it, but it says it's a nociceptor. Nociceptors are for deep pain. They're for pain that are deep. But just a regular nerve ending comes up to the top where you can feel things. This is called the Pacinian corpuscle. It senses pressure if someone's just poking you. The Meissner's corpuscle senses some sort of touch right here. It looks like some gentle touch right here. We have thermoreceptors that sense cold or heat, and then pain for our nociceptors right there. So here's something being poked right here, something being pinched, that kind of thing. So quite a few in there. By the way, this the sense of touch is considered a simple sense, where the other ones are considered complex. But look at all the complexity here. Lots of different things here to help us learn to feel things, right? So it turns out it must be important for us to be able to feel things so that we can respond. Remember, one of the criteria for life is sensing and responding to your environment. Part of that, of course, is to keep us safe. If we're being hurt, we need to pull away from that hurt so we don't damage any tissue. There is such a disease where people are born without the sense of touch. Yikes, right? And so I know that there was a story about a child that didn't have any sense of touch, and the parents figured it out when I think they went to go spank the child, and this child didn't feel it, and so it didn't help at all. Furthermore, the child burned himself frequently because he didn't know he was touching something hot. He broke bones and continued to walk on broken bones, and it made them worse. So it's a terrible thing to have, and you definitely want your sense of touch. That would be an awful thing to live with. That particular family had another child, and it turned out also had no sense of touch. Yikes. Very rare, very rare. Okay, the sense of hearing is, of course, done by our sensory organ called our ear. And let's take a look at it. Turns out we have the 3 smallest bones in your body are in your ear. You're supposed to write this down. They're called the hammer, the stirrup, and the anvil. They have actual names called the incus, the malleus, and the stapes. But for now, I guess in our book, we're using hammer, stirrup, and anvil. This right here, the tympanic membrane, is the eardrum. And the eardrum begins to vibrate and vibrates the hammer, which hits the anvil, which hits the stirrup, which hits the foot plate, which joins up to the inner part of the ear, which we'll get to in just a minute. So it's all about vibration. Sound is vibrating. My voice is coming out through my voice box right now, and it's vibrating through the air. It's going into my microphone, and then the microphone's coming into your speaker, and that is being interpreted and sending out a vibrating sound through the air to your ear. And as that air gets to your ear, it vibrates the eardrum, the hammer, the anvil, and the stirrup. Okay, so here it is. I'm talking, it's coming into your ear. This part of your ear called the pinna, sometimes called the ear leather, and dogs especially. This is functioning as a funnel that's funneling the sound into your ear. So you may have noticed, if you can't hear somebody, if you cup your hand around your ear and essentially make your ear bigger, right? That's going to bring more sound into your ear. So you can see how that funnels. So for instance, if I lost that ear for some reason, I don't know, like Vincent van Gogh got my ear cut off, right? I can still hear because the hearing is not done by that part of your ear. The hearing is done in here. Okay, so the sound comes in through the ear canal. There's the eardrum, the tympanum. It's going to start vibrating. There's our 3 bones. Okay, and there's that oval window right here. Okay, so this is called the outer ear. This is called the middle ear, and this is called the inner ear. All right, so in the outer ear, we get the sound coming through air, gas. In our middle ear, we have the sound vibrating through gas. So there's air in here, right here. And in fact, sometimes this little tube will close up, especially if you're going up in a plane or up a mountain or something, causing it to make your ears kind of, feel like you can't hear very well. And if you blow your nose or chew a piece of gum or swallow, you'll open up the eustachian tube and get that air back in there and pop the ears back open. That's what's happening, the air in this part right here. Well, then once the vibration gets to this is a complex organ consisting really of 3 things. We have our semicircular canals right here, our vestibule right here, and the cochlea. Cochlea means snail. Looks like a snail cell. It's actually made of bone, and the bone, so it's hard, but it's coated on the inside with hairs. Once again, we have some auditory hairs that are in there. I will tell you, let's see what our next slide is. Okay, that's the cochlea. All right, so before I get to that, I want to tell you this, right? The semicircular canal, so instead of air in here, this is filled with fluid, okay? And therefore, this fluid is traveling, the semicircular canals, we have one going up, one going out, and one going this way. So the three, like X, Y, and Z axes right there. There's a little patch of tissue in there called the macula. And what happens is when you tip your head to the side, the fluid moves through the semicircular canal and causes there to be this little rock, so like a little stone, it's called an otolith, to move past little hairs, and that tells the body, it tells your brain that you just moved your head to the side. Okay, so your brain knows that you moved your head to the side. Okay, then you bring it back up, move it to the other side, and your body knows that. This is important because this is how we balance. All right, our brain, it needs to know where gravity is all the time, and it does that by registering that through the vestibule and the semicircular canals. So these two parts of the three parts are dedicated to balance, and that's why we've turned balance into one of the senses. All right, but the sense of hearing happens all in the cochlea right here, which is our next slide. Okay, so hearing takes place in the cochlea. Remember, it means snail. Now, we've unwound it a little here to make it a little more understandable. It's a fluid-filled tube. It has tiny hairs that line it. They're sensitive to various frequencies of sound. All right, so the sound comes in. Here's that round window. The oval window where that stirrup was. The stirrup was hitting right there. Vibrations come through. So sounds coming here. If we have a wavelength of 7,000 hertz, we can hear it right here. It travels through. All right, and we're hearing different frequencies of sound. Here we're at 200, woo, and then coming back down to 400. So, and back around. 1,500 hertz, 2,000 hertz, all the way dissipating sound, and comes back out to the round window. Okay, so it's like traveling through that tube. All right, and at different areas is when it's hearing different frequencies. High frequencies down here, lower frequencies in here. Okay. All right, pretty cool. There is, if someone is deaf, if someone has lost the ability to hear, some of the times it can be because of the cochlea. Sometimes the fluid is not fluid enough. In other words, it gets real thick fluid. The hairs don't move around very well. And those people can possibly hear again through a process known as a cochlear implant, which is essentially trying to mimic the cochlea by putting in a stimulator that will take in frequencies and interpret that to the brain. So that's been very helpful for some deaf people out there. I'm going to take this back to here real quick and tell you just a couple things else about this. I have a great story about a student of mine who was in my general science class many, many years ago in Austin. And this kid was in class and he told me, well, Mrs. Casper, I'm not going to be here next week because I'm having surgery. And I said, oh, wow. His name was Samuel. I said, Samuel, what's the deal? He goes, well, when I was very young, I had an ear infection. And so, and the ear infection is when fluid or pus actually fills this middle ear area. I don't know if you've had an ear infection before. My daughter had them all the time, and we ended up putting a little hole in her ear called a tube in her ear. It goes right there through the eardrum and lets this fluid that collects in here just leak out so it doesn't build up and cause damage. Well, this kid, Samuel, had this fluid in his ear right here, and because of the nature of it, for some reason, it actually dissolved these three bones. It dissolved them. And so therefore, if they didn't make the connection, he couldn't hear, right? And so his other ear was fine, so he could hear, but this one ear wasn't. So he said, well, I'm going into surgery, and they're going to see if they can replace this. So 2 weeks later, he came back. They had put something in here to resemble these bones, and he had hearing in that ear. I think that's a fascinating story. I tell it to all my students. Okay, this is going to give us a little more insight into hearing. Let's listen.
The ear has three parts: the outer ear, or auricle, the middle ear, and the inner ear. The auricle, the visible part of the ear, collects sound waves and directs them to the auditory canal. The auditory, or ear canal, is lined with cilia and ceruminous glands. The earwax from the ceruminous gland and the cilia protect the ear from entry by foreign matter. The thin tympanic membrane separates the outer ear from the middle ear. Sound waves cause it to vibrate. The three tiny bones of the middle ear are the hammer, or malleus, anvil or incus, and stapes or stirrup. These bones pick up the vibrations from the tympanic membrane and transmit them to the inner ear. The eustachian tube of the middle ear opens into the pharynx and permits air pressure to equalize between the ear and the outside air. The inner ear is maze-like, consisting of bony and membranous structures surrounded by fluid. The semicircular canals are fluid-filled bony loops that help maintain our body's balance. The cochlea is a fluid-filled, snail-shaped structure that houses the organ of Corti, the true organ of hearing. The fluid aids in the transmission of the vibrations. The organ of Corti changes the vibrations into nerve impulses that are picked up by the auditory nerve. Sound waves are collected by the auricle, or outer ear, and sent to the auditory canal, tympanic membrane, ear bones, and then to the cochlea, where the vibrations are changed by the organ of Corti to nerve impulses and transmitted by the auditory or cochlear nerve to the temporal lobe of the brain for interpretation.
Okay. Once again, the brain also will have memory. For instance, right now, you're hearing my voice. You know it's my voice, right? You remember my voice. You remember your parents' voice. You remember a song, a singer, this kind of thing. So that's how complex our brain is, right? I do have the ear balance in here. So this is the semicircular canals. Remember, they're going this way and then that way and the other way. So the cochlea is over here. This is all filled with fluid. So the vestibule has this little thing called the saccule and the utricle, and this little part right here. So that's where the little stone is rolling over this. It's called the crista ampullae. And then the perilymph is going, so it knows if your head is leaning to the right, leaning to the left, leaning back, that kind of thing. And it helps also with our cerebellum, with our balance. You know, that's helping us with our coordination, and in conjunction with where that fluid is in our ear helps us keep our balance. Pretty cool stuff. Take care of your ears. Can your hearing be damaged? Yes. Exposure to loud noise can cause damage to the hairs in the internal structure. And therefore, how can you take care of your sense of hearing? You know, y'all probably listen to those little earbuds. You stick those earbuds in there. Just try not to listen to that music too loud, too long, okay? Because it can cause damage over time. In fact, some of those great rock and roll musicians from the old days, you know, they're having hearing problems right now because they listen to loud music and played loud music all the time for a long period of time, and it will damage that. Furthermore, just with age, we tend to lose some of the flexibility of the cochlea and the ability to hear as well. I know, for instance, as we get older, those higher pitches are more difficult for adults to hear. Okay, so our lab, the things we would have done today if we were together, I would have made you taste something salty, sweet, sour, bitter, and umami. You just put a little Q-tip in there and taste it and see what you thought. And that would have been lemon juice, sugar water, salt water. Bitter, I used something called tonic water. It's kind of got a bitter taste to it. And then for umami, I would have put in some Worcestershire sauce. So you can try that at home if you want to. Also, you would have guessed a scent. I would have given you a little something to smell. It would have been one of my essential oils, you know, to see if you could smell. Something like lemon, peppermint, things like that. Now, the touch experiment, like I mentioned earlier, is the one that's in your book, so you can try that if you want to. Maybe get a sibling or somebody to help you with that. And then in a second, we're going to do our eye dissection. With the ear, as far as balance goes, because that inner part of the ear, the vestibule area, the cochlea area, are filled with fluid, if we get fluid moving, it doesn't stop as very easily. Think about if you've ever been in one of those round above ground pools and you walk around in a circle for a lot and a lot and a lot, and you stop and it just carries you, right? Because that fluid will hold on to that motion. Same thing goes in your ear. If you spin around, the fluid starts moving in that ear, and when you stop, the fluid keeps going, and that's called dizzy. Yeah, that's how you get dizzy. So you can try spinning around if you want to, maybe on some of these fun days outside, go make yourself dizzy. All right, now I'm going to play this video of me dissecting this eyeball. One thing I want you to do is make sure to observe safety at all times. Remember, that blade is very sharp. You need to be careful. One thing you must always do is always to cut down onto your little plate, okay? Don't ever hold a specimen in your hand and use the blade, because the blades are very sharp. They could actually go right through the specimen, right into your hand, and we don't want that to happen. That glove is not going to protect your hand from a blade, okay? So I need you to be very careful that way. Have y'all, I don't even know if y'all have used some of these blades yet. Have we done any dissections yet? I don't remember what we did so far. So just be very cautious, and you can watch the way I do it and then go try to do it yourself with your own eyeball. Okay, here we go. All right, it's time for the eye dissection. Okay, so I gave y'all a little plate like this, and that's okay. You can put that right on the plate right there. And I'm going to use my dissection mat. Okay, so put your gloves on. Get your eyeball, cut it out of the bag. Okay, keep the bag because you're going to put this back in at the end. Okay, so first thing we're going to do is going to take a look at it. Pretty creepy, huh? All right, here, this would be the optic nerve that's coming out right here. Okay, so this would be the spot on the eyeball on the inside where the blind spot would be, right, for that eye. Notice there's a lot of muscle here that got cut away because the eyeball needs to move around a lot, right? So there's a lot of very intricate muscles right there. And then we have the tunic. So yes, it's a little gooey. Yeah, I will confess to you that eyeballs gross me out just a little bit because without the fluid inside, you know, they're flat. So that kind of creeps me out a little bit. All right, but you can see right here, it is got a cloudy lens right there, and that has to do. Sorry about this, it's a glitch. It'll come back. Whatever that is used, if we had a fresh eyeball, that would be nice and clear. Okay, so once we get that open, we're going to see what's behind there. All right. This one has a lot less tissue on it than some of them do. Oftentimes I'll have it with the tissue over the top that even has eyelashes coming out, so that's kind of gross. Okay, remember you're going to use your very, very sharp blade. You're going to be very, very careful with this, okay? And what I've devised over the last few years is to cut open this way. All right, so I'm going to separate the top from the back, okay? So hold it kind of sideways like this. Remember, always cut down onto your plate. Get a nice grip right there. And you're going to find out how tough this tunic is. You have to really cut into it. You have a very sharp blade. So we're going to cut in. Now, hopefully it didn't squirt out at you, Erin. Hope not. All right, so it's going to come. We're going to go ahead and cut all the way through. You should see some liquid, and it'll start coming out pretty soon. And it's kind of a gelatinous liquid called aqueous or aqueous humor and vitreous humor. All right, so there's some of it coming out. So see, I'm just making a round cut all the way around the tunic. It got real thick right there. All right. Okay, so now we can see the inside right there. All right, so we'll get to this in just a second. Let me go ahead and cut this part off. Okay, so this is the front of the eyeball, okay? So you can see there is a clear spot right there, okay? That's the pupil, okay? That's the part that we see through, the light comes through. So the part that's around it right here is the iris, okay? So the iris, see those striations on there? Okay, that's muscle. Because remember, the iris will close and open the pupil depending on how much light wants to be let in there. So that's a muscle, but it's also the thing that shows the color of the eye. So here we're looking at a sheep eye. I'm guessing the sheep had rather dark eyes, brown eyes right there. Okay, so then this is see it's clouded over right here, but it would be see-through. What's interesting is that this tunic right here that's white is the same material as this part right here. It's just this part's clear. How about that? But it's that same tough tunic right there. So that's the cornea, and the cornea is what we look through, okay? It does serve a little bit of lens activity. In other words, it helps to bend the light just a little bit. Okay, now we're going to get into the more interesting parts right here. So you can see there's some goo holding everything in place. And one thing you're going to do is take that goo out, and you're going to peel it off this little hard thing right here. So this is the vitreous aqueous humor. Yours might not be as thick as mine is, okay? It might be more gooey, gelatinous. I'm not sure. Some of that iris got stuck on there, it looks like. Okay, so now we're left with this, okay? This is the lens. All right, once again, if this were a fresh eye, this lens would be completely clear, and it would look like a little magnifying glass. And in fact, if you put it over some words or something in a newspaper or whatever, it would magnify the words. Okay, so that's the job. Whoa, hello, it's a little slippery. That's the job of the lens, is to direct the light and to focus it. So I'm going to squeeze it a little bit. Try squeezing it. It's very hard. It wouldn't be this hard if it were fresh. Okay, it'd be a little more malleable, we could squeeze it a little bit. But you see here, it's shaped mostly like a circle, but a little bit oblong like a lens would be shaped. Okay, so that's our lens. There's our aqueous humor. So here we're going to clear this stuff away from the back here, some of that aqueous humor out of there. All right, remember your fingers are a really good tool for dissection, so I'm just going to take some of that out. And then I want you to see there's this part right here. I don't want to cut it. That's sticking out from part of the eye right there. So that's, do you see where it's sticking through right there? Maybe on yours you can see it better. Right there. That's where the optic nerve comes in. So there's your blind spot right there. Okay? I'm going to scrape it away now. Okay? Because what I really want to see is this really, really cool retina. So this part right here is the retina. So what I'm going to have you do is just gently kind of peel that away. You really should do that down here so you don't cut yourself. Okay? Bad technique, Mrs. Casper. All right, so you get a little spot like this. You can actually peel that off. Mine's being a little stubborn today. Look at that beautiful color. So it's got black, but almost like that kind of pearlescent look there. I wonder if maybe tweezers would be useful right now. Usually this rips off pretty easily. This guy's holding on to his retina. All right, there it is. Okay, so there's the retina. See, it's got that beautiful sheen to it. There's where that optic nerve was. And look how thin it is. On the other side, you can see it's completely black, like my glove, right? And that little thin strip of tissue right there has those fancy rods and cones in there that are receiving those light signals, those light stimuli, and taking all of it and interpreting it to your brain. Isn't that amazing that that simple, small thing right there can do all that complicated, I'm not holding it, that can do all that complicated vision. That's pretty amazing stuff right there. And I happen to believe it's just beautiful. Very nice. So once you've taken that away, you can see now the back of the eyeball. You can see where that optic nerve was coming in right there. Okay? And once again, you can feel how tough that tunic is. And that shows you because the eyeball needs to remain really tough, right, to protect it. Okay, so really, gang, that's it. That's all there is to it. There's not a whole lot, but we saw the interesting parts right there. So our cornea, our iris, our tunics, our retina, and our lens right there. Okay, very good. Congratulations if you got through this dissection all on your own. Well done, y'all. Okay. Okay, so now when you're finished with it, you can collect the parts, put them back into the bag they came in, and put that bag back into the Ziploc bag, right? And throw that away. You can throw it in the trash. It's okay to throw it in the trash can. All right, and then when you wash your blade, be sure to wash it very carefully. Remember, it's still very sharp. And then let it dry without the cap on it. If you put the cap on it, the water will be trapped in there, and it will cause it to rust pretty badly. So leave those apart in a safe place. Hopefully you don't have any younger siblings. And then you need to put that, give that to your parents or whatever. They can tape it up, you know, like put the lid back on it, tape it up, and throw it away if they don't want it around. Wash it. If they wash it with soap, it's a useful tool to have around. You know, I keep them around for various crafts and things like that. All right, and then wash up. You just throw your plate away with everything out like that. All right, students, I hope you enjoyed that. And what we'll do next week is we're going to go back to Module 15, remember, and start looking at the immune system, the urinary system, and one other, lymphatic system. Okay? So I will be back with you next week. Very good. Bye.